Oh, by the way, I remember that in this class, last week, there's a student who asking about why in RNA we have uracil instead of thymine in DNA, right? So I checked for the research on PubMed. So it turned out that the T in DNA is more stable because it will quite resistant to the photogenesis mutation in the nucleus. So with the thymine in the DNA is more stable and maintain the function of DNA is to storage and duplicate the information. However, if the thymine is go to inside outside of the nucleus, so this is the nucleus. If the thymine is go outside nucleus, so the thymine is quite stable inside the nucleus, but when it gets outside in the cytoplasm, it's quite susceptible to the oxidation in the cytoplasm. So as a result, the thymine will be easily to be destroyed outside of the nucleus region. So that's why the RNA must use a similar structure, uracil, which is more resistant to the oxidative in the cytoplasm. And of course, the uracil is not quite stable to the photon mutation, but it's quite stable to the oxidation. So the RNA, because the RNA have to move out of the cytoplasm, so uracil could be the better material. Okay. And in last week, we have discussed also on the promoter of the prokaryotic cell and the promoter of eukaryotic cell, and we discussed, on the differences in the DNA replication and RNA transcription. So in here, DNA replication using both templates as a strand, both DNA strand as a template to synthesize the new DNA strand, follow the semi-conservative rule. And in transcription, it's used only one template, only one strand as a template. So the product of the DNA replication is double-strand DNA because it's using two DNA double strand as a template, and in here is a single strand RNA because only one strand was used as a template. So the substrate in here is deoxy nucleotide, and in here is ribose nucleotide. The primer DNA replication require the RNA primer. Meanwhile, the RNA transcription requires no primer. Okay, so the enzyme participation process in here is DNA polymerase, and in here is RNA polymerase. So we discussed also on RNA transcription. So in here, it using only one DNA strand as a template, which is the sequence from 3' to 5' here. So the RNA polymerase will binding on only on this one, and that's why we call this one is a template strand. The other sequence here, the other strand here, will have the sequence the same with the mRNA. So replace only the T thymine by EU uracil. So we call it the DNA coding strand because this strand have the same sequence with the mRNA here. And sometimes we call it the positive strand. Okay, and the other strand here is used as template. We call it the negative strand. Okay. Wait. So come to today's lecture. So first, let's discuss on how genes were structured in the prokaryotic cell. So in here, you can see that this is the promoter regions. Oh, promoter. And this is the position +1. +1 is also called TSS, which stands for transcription. Starting site. Transcription starting site. So the promoter will binding in here by recognize these two regions, is binding here, and slide until it reach the +1, the TSS, and it start to synthesize the mRNA here. So this is the same between the prokaryotic cell and the eukaryotic cell. But the difference is in here, several genes will organize together as a structure called operon. So operon is several genes which have a similar function that encode for quite a similar protein, or the protein with the same similar functions that share structure organized as a structure called operon, and they share the same promoter. Okay, so whenever the operon was activated, so the transcription factor binding in here, and it will transcribe the whole mRNA, which is encoding for different unit of protein. Okay, so genes here is defined as a unit for a unit of protein. So in here we have different genes organized as an operon. Let's watch a small clip to this demonstration on this process. So this is the nucleus region of the prokaryotic cell. So fragment. So this is the two typical reason that the promoter, that the transcription factor recognize this is the promoter is have binding in the -10 with the typical TAT sequence, and the 30 -35 with the TTGACA sequences. Okay. Okay, so transcription factor binding in. Okay, so again, you can see that in here, the now strand, the DNA strand in the below here is called the DNA template strand or the negative strand. Okay, so this, the sequence here is the non-template strand or the coding strand of DNA, or the +1, the positive strand. Okay. Okay, so the promoter binding, the transcription factor binding in promoter, but it not start to synthesize the mRNA right there, but it slide until it reached the position +1 or the transcription starting site. Here is a transcription starting site. Okay, and it starts to synthesize the mRNA from this position. Okay, so the mRNA here. The mRNA was synthesized, follow the Chargaff rule. So you can see that in prokaryotic cell, the transcription and translation continuously processed. So as soon as the transcription had not finished yet, the ribosome have come and binding into the mRNA to start the translation process. So this is a continuous process. Transcription and translation would happen at the same time. So as long as the transcription factor reach the termination region, it will release the mRNA and the transcription process finish in there. So come to eukaryotic cell is a little bit different. Where's my? So it's the same vision like prokaryotic cell, that it including the promoter and the transcription factor when binding in here. Transcription factor. And it's lie until it reached the position +1. So this is a transcription starting site. But different with the prokaryotic cell, that in eukaryotic cell it has the region called the intron. This is different with prokaryotic cell. So as long as the premature mRNA was synthesized, it's including all the region, but it had to been through the process called splicing. Splicing to remove all the intron region here and put the 5' cap and the poly A tail in here. So the typical structure of the mRNA is in the slide you can see here, with the 5' cap, the 5' UTR, the coding sequence. Coding sequence in here is coding for protein. For protein. And 3' UTR. So maybe I will open a paint file to explain more clear. So this is transcription and translation. So in here we have a double-strand DNA that encode for genes. Double-strand because it's encoded for gene is will have its promoter. So let's hypothesis this is a promoter region. So this is and as the promoter region, the transcription factor will binding in there here. With binding in here and start to synthesize. But it will not start to synthesize the mRNA from this position. It starts in here, in the position +1. This is position +1. Stand for TSS. So different with the gene in the prokaryotic cell, in eukaryotic cell we have intron. Here is intron. We have an intron here. Okay. So the transcription factor binding in promoter region slide until it reaches the position +1 and it start to synthesize the mRNA here. So this is the premature on A mRNA with the sequence of intron. Okay. So this is translation. Sorry, it's transcription. And the premature mRNA will been through a process to become mature mRNA with the intron was removal or splicing and the 5' cap putting here. This is 5' cap. And poly-A tail. So this poly-A tail, that means it's a, and thanks to this poly-A tail, whenever we want to catch the mRNA to investigate, for example, the transcription level of a specific gene, we will put in there the primer, including the poly-T, the oligopoly-T, to catch the poly-A tail. So in here, the intron was removable.
Excuse me, can you tell me again what does the poly A tail do?
So the poly A tail in here during, I don't know if I should. So the poly A tail here is the the structure of poly A. So it including many A, and it attacks into the 3' direction of the mRNA. Okay, and in order to catch this this mRNA to investigate, for example, a transcription level, the scientists will design the oligo T to have it binding with the poly A tail to catch the mRNA. Okay, it's a structure formed during the process of the mRNA was mature. Okay. Right. So this poly A tail, and the intron was removable. Remove. Nice. And during splicing process, different exon will be removed too, but we will talk about it later. So this is the mature mRNA. So it's going to translation process. Translation. So that means that so the position +1 will be maintained in order here. +1. So as a consequence of the translation, the ribosome, so this is 2 subunit of ribosome, will come and binding in the mRNA and start to synthesize the polypeptide. But like the transcription factor in here is binding in promoter region, but it slides until it reaches the transcription starting site. So ribosome will binding in here, but it will not start to synthesize the protein immediately. It will slide until it reaches the start codon. For example, this is the start codon. Start codon is AUG, which is encoding for methionine. So from this position, it will start to synthesize the polypeptide. So the first peptide is methionine here, and we'll continue to start to synthesize another protein, another amino acid here. Until it reached the stop codon, right? So in universal coding sequence, we have the stop codon is UAA or UAG or UGA. Okay. So in this position, the polypeptide was stop synthesized and it released and going to is the post-translation modification. But in here, we have the regions from the position +1 here into this. We have the sequence, the region here from this point to this point. Okay. So the mRNA, which is the transcription starting site, was in this position, but the ribosome will not start to synthesize the polypeptide until it reaches the start codon. So this is the translation starting site, and this is the transcription starting site. And this region of mRNA was transcribed but not translated. So that's why we call it the 5' UTR. Oh, maybe I will choose another column too. 5' UTR or 5' untranslated regions. Okay, so the position from this to this, and the same with the position here from the start codon to the end. So we have the region here. This region was transcribed. It's located in the mRNA, but not translated because this thing is not coding. So we call it the 3' UTR regions. Okay. So like the structure of the mRNA here, we have the 5' UTR and the 3' UTR here. So this is the position of +1, the transcription starting site, and this is the start codon. All the region in here is 5' UTR. So some of you have participated in the bioinformatics practical training. Do you remember the exercise number 5 here? So in exercise number 5, they want us to explore a gene called MYH9, right? And in question 3, they want us to look at in the transcript table in the transcription of MYH9 201 and see which exon is completely and partially untranslated. So to explore that, we go to MYH9. So for the student who have not attended to the practical course, you go to Ensemble, which is a very large genome database. Click into human, explore the MYH9, click on go. You click on the human genes, and here we have observed different transcript variant of the MYH9. What is a transcript variant? We will discuss in a few minutes. But for the moment, just focus on the transcript MYH9 201. Okay, you can click in here. Oh, it don't provide a picture here. Maybe YouTube. Okay, so the MYH9 201 in here, it including as I remember 41 exon. Yeah, 41 exon. But among those, only 40 was coding exon. Okay, so we can see in here in this figure, it including 41 exons. So the large particle, small but thick particle here is exon, and the small and thin line here is the intron. Okay. And if you observe an exon with a dark color in here, that means that it encoding for protein. And the blank exon here, the square with a white blank in the middle here, is the non-coding region. So we can see that all the exon in the middle was coding for protein, but we have a part in the beginning here is not coding for protein, and the part in the end of the gene here is not coding for protein. So because this is the 5' UTR of the genes, and this is the 3' UTR of the genes. So that means that this region,
excuse me.
yeah.
So are there the big, there are the large vertical part, are they the intron?
I'm sorry, what?
Are there the intron, like the brown thick part and vertical part?
Yeah, I just explained. So this small line here is the intron. So here is the intron. Here is the intron here. Okay. Do you see my point stick?
Okay.
Yeah. So this is the intron, and this small here is the exon. Okay. Exon. Part here exon. Okay. You see it? Okay. So all the fragment here is the exon. Okay. Okay for you to recognize. So in here we have 41 exons, so it's a very large gene. And the part of here is not coding for protein. That means that this is the 5' UTR, and this is the 3' UTR. Okay. And in the middle here is the CDS. That means the coding sequence. Okay. Right. So one phenomenon is quite interesting also is the alternatively splicing in prokaryotic cell. So that means that not all the time all the exons was transcribed into mRNA. So for example, in the promoter here, so this is transcription starting site. We have a gene with 4 exons, exon number 1, 2, 3, and 4. So in one way of splicing, the mRNA will be synthesized with the sequence from all the exons, exon number 1, exon number 2, 3, and 4 here. So it's a classic splicing. But in alternative way of splicing, where the process somehow overskip the exon number 2, so the new mRNA now have include only the exon number 1, number 3, and number 4. So this is called the alternative splicing, and it's called to the transcript variant that we have just observed. So we just observe in the MYH9 here, we have so many transcript variants. We have at least 6 variants is coding for protein, and the rest is not coding for protein. Okay. So how is so? So another example in here, we have a gene with 1, 2, 3, 4, 5, 6 exons. And the splicing was including, because of the different way in splicing, in the one type mRNA, we have including only exon number 1, 2, 4, 5, and 6. Somehow the exon number 3 was skipped in this type of translation. And as a result, the protein number 1 including only the region from 1, 2, 4, 5, and 6, but not 3. Another way is to the mRNA was splicing based on exon number 1. It's overskip exon number 2, 3, overskip the 4, 5, and 6. So as consequences, we have the protein including only 1, 3, 5, and 6. And in the another way of splicing, we have only the 1, 3, 4, 5, 6, and the 2 was skipped. So like in here, we have different ways of splicing. So in among 6 transcript variants that encode for protein, the largest one is the 215 here, okay, and it's encoding for a protein, very big protein, 1981 amino acids. Meanwhile, the smallest one variant is coding for only 103 amino acids. So you can see in the table the relative size of the protein here. So the largest one is the 215 here, and the smallest one. Okay, somehow it's overskip all the 1, 2, 3, 4, 5. So this one is including only 3 exons, 1, 2, 3, and this one is including nearly 41 exons. Okay. And the higher vertebrate, the higher level organisms, the more way of alternative splicing is might have. So scientists have found out in Drosophila, the DS-CAM1 gene have over 38,000 transcript variants. So that means that this gene was encoding for more than 38,000 transcript mRNA. So this is research published on Genome Research, have found out that in the human genomes, in 25,000 genes, human genome is composed of 25,000 genes. Only about less than 5,000 genes was encoding only for one mRNA molecule. About nearly 3,000 is have 2 transcript variant, and so on. So more than 2,000 genes have have 3 transcript variants, and so on. So about 1,000 genes will have more than 10 variants. So some genes has many different variant transcription. So for example, like in this gene, the gene is the FGFR2 genes. This gene was encoding for a cytokine that played a role in the embryonic development. So it's a carrier having a mutation in these genes. It can lead to the abnormal in the development of the skull and the skeleton during embryonic development, and led to consequences that the carrier could be intellectual disability when he or she was born. But anyway, you see that this gene is another really big genes, and including many different exon here. So the first exon in here is the 3' ETR actually. In here we have different transcript variant of the genes. So for example, we compare between these two one. So the exon here is including in this variant, but somehow it was lacking in the second variant. Meanwhile, this 2-1 here will exist in this variant, but somehow lack in the other splicing variant. So let's watch a small clip demonstration about RNA splicing. I don't know, remember if I have shared the sound.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
Okay, so now we will have a small break, a 5-minute break before we come to another part, translation. Okay, 5-minute break.